It's 1664, and this guy, Robert Hooke, is messing around with his new toy, which is one of these. Yes, you guessed it. It's a microscope. One of the very early microscopes, pretty much just been invented. Hooke was a very keen artist from being a child back on the Isle of Wight. So eventually, in 1665, he published this book, Micrographia. Some of the observations that he made during the writing and, well, drawing of this book, and his fascination with fossils, started him down the path of being one of the forefathers of Darwin's theory of evolution by natural selection. So that's now 200 years before Darwin. Robert Hooke started laying the foundations for it all, realizing that things were changing over time. Here's the picture that kicked off a branch of biology called cytology. The drawing is of a section of a piece of cork. Now, you see those little spaces in there? He called those spaces cells because, to his mind, he thought they looked like the cells monks slept in at a monastery. And this, my friends, was the very first step into cell biology. Now, as it turns out, there are two basic types of cell. Anybody who's thinking now, plant and animal, all right, you've got your prokaryotes and you've got your eukaryotes. Now, remember this, because this is important. Prokaryotes don't have a true nucleus. Okay, their nuclear material does tend to be concentrated in one specific area, but it's not surrounded by a membrane or separated from anything else. And most of this world's prokaryotes are bacteria, and there's archaebacteria as well, which are an even more ancient sort of organism. Now, the eukaryotes, which includes us, do have a nucleus. In fact, that's the reason they're called eukaryotic. It all comes from ancient Greek. Karyote is ancient Greek for either nut or kernel. There you go, that's where the name came from. Something else that separates eukaryotes from your prokaryotes is that the eukaryote have got distinct organelles surrounded by a lipid membrane. And, all right, yeah, I know what you're thinking. You've got that picture of general plant cell, general animal cell, which look like this. And you're thinking, yeah, organelles, yeah, I know what they are. So you got your nucleus, you got your mitochondria, you got your chloroplasts, you got your vacuole, and all that gubbins, right? Well, sorry to disappoint you on this, but it's a fair bit more complicated than that, I'm afraid. And this is where electron micrographs come in. Here is an electron micrograph of an animal cell. Yeah? Now, here's one of a plant cell. A lot more to it, isn't there? You're going to need to be able to recognize these structures just by looking at electron micrographs. What we're going to do is go through what these pictures actually show. Now, what I'm going to show you first, though, is the same pictures but with false colour, so you can pick out the different structures. And then, as I said, we'll go through each one and what it does. So breaking it down then, we're going to look at the basic structures in an animal cell and what they do. Then we'll look at the structures that only occur in plant cells and have a look at what each bit does. Now, first up, we'll start with, well, back in the old days, GCSE level 2, you would have called this cell membrane. Well, now because there's quite a lot of cell membranes, we're calling this one from now on the plasma membrane. it's more complex than it was first thought. And I mean, it's going to get its own full video along this sort of lines. But basically, what you've got is a lipid bilayer, a double layer of lipids with hydrophobic ends in the middle and hydrophilic ends on the outside. And that's kind of punctuated by proteins or channels like going part way through, all the way through. And there's sort of carbohydrates and all other kinds of stuff sat on the top surface. What's it for? Well, this really goes all the way back. It controls the movement of substances in and out of cells and organelles. It's also involved in cell adhesion, so cells sticking to other things, ion conductivity, obviously, because, you know, controlling things going in and out, and cell signaling, because, yeah, cells do kind of, well, I won't say talk to each other, but, you know, they do communicate. Not only that, but the plasma membrane serves as the attachment surface for structures such as, if you're in a plant or in a fungus, the cell wall. Also the glycocalyx and the cytoskeleton. And we'll talk about those at a later time. Next, one that you've heard of, still, the mitochondria. Now, as you know already, the mitochondria is where the energy is produced. And again, this is something we'll go into more detail in a later video. Inside the structure, you've got things called cristae. You see those sort of shelvy type things? That's them. Now, they are absolutely covered in the various enzymes that are involved in releasing energy from sugars and oxygen. Like I said, we'll hit that one a lot harder in later videos. So we're talking glycolysis, Krebs cycle. They are also involved in cell signaling, cellular differentiation, controlling the cell cycle, cell growth, and cell death. Now, interestingly, with these mitochondria, the DNA, because there is DNA inside the mitochondria, it's inherited directly from your mum. Now, that has got its uses, certainly in evolutionary biology, because we can use that to see how related populations are, and in terms of molecular biology, certainly, it's one of the biggest pieces of evidence in favour of evolution, because once you've found these populations and how related they are to each other, you can then use the information you've got to link back to other populations. Next, bit of a mouthful, the rough endoplasmic reticulum. In terms of structure, this thing is made up of flattened sacs covered in ribosomes. And the ribosomes is why it's called rough endoplasmic reticulum. Because if you look on the diagram later on, you'll find that there is also smooth endoplasmic reticulum. Now, its membrane, if you look, is actually attached to the nuclear envelope. That's because the function of the rough endoplasmic reticulum is to synthesize and fold proteins that are translated from the DNA. Now, the translation happens, and transcription is down to the ribosomes, and the DNA itself is stored in the nucleus. After these proteins have been built, they are shipped off to the Golgi Apparatus, and we'll talk about that later on. Now, smooth endoplasmic reticulum then. Now, this is a little bit different, because instead of it being flattened sacs, the smoothia is made of cisternae. Now, the main job of this organelle is storage and synthesis of lipids and steroid hormones. Now, it turns out that it also stores and metabolizes calcium products and is part of the mechanisms that detoxify harmful metabolic byproducts. Proteins, lipids, hormones, all made inside the endoplasmic reticulum, either rough or smooth. So how do they get out of the cell into your bloodstream? The answer lies right here in the Golgi apparatus. Now, have a look at the ends of the structure. Can you see those little spherical things that are kind of budding off it? That is how stuff that is made in the endoplasmic reticulum actually gets out of the cell, or around the cell, depending on what it's needed for. Now, the Golgi apparatus, which is sometimes called the Golgi body, modifies the structure of products, concentrates them, and puts them in vesicles. which can then be moved to another part of the cell, or out of the cell, or to join up with the plasma membrane. Now, yeah, secretory vesicles, that's pretty self-explanatory. What you've got is a substance with a lipid bilayer wrapped around it. Now, another thing that is made using bits of Golgi body are lysosomes. Now, lysosomes, if you look down there, they are spherical vesicles that kind of, well, they've got kind of a soup of hydrolytic enzymes inside them, which are capable of breaking down just about any biological molecule you could name. It's kind of what's in biological washing powders, except if you tried putting lysozyme, which is the enzyme soup, into your washing, it wouldn't just take out the stains, it'd take out your clothes as well. These lysosomes are involved in quite a few different cell processes, including secretion, plasma membrane repair, cell signaling, and energy metabolism. Their main function, though, looks to be, and seems to be from observations, the digestion of unwanted materials in the cytoplasm, such as waste and obsolete components. Now, let's take a look at the last couple of major structures, the nucleus and the centrioles. The nuclear membrane, simply enough, separates the nucleoplasm, which kind of contains DNA sort of all over it, as long as you're still in interface, from the cytoplasm that kind of gives structure to the rest of the cell. The membrane is actually a double membrane made up of two phospholipid bilayers. Now, the outer membrane is attached to the endoplasmic reticulum, and the inner membrane is covered by a mesh of filaments that stabilize the membrane as a whole and are involved in how chromatin works. And chromatin, if you remember, is part of cell replication. So another one, we'll get onto that in a later video. So these two membranes are actually interconnected by nuclear pores, which run from the nucleoplasm out into the endoplasmic reticulum. So basically, DNA can move backwards and forwards. And the breakdown of the nuclear membrane is the first sign that interphase has finished, and you're about to move into cell replication. Now, you see the dark bit in the middle of the nucleus? That is the nucleolus. And not to, well, just kind of, it seems like I'm glossing over it, but it's got one simple function. It makes ribosomes. And without ribosomes, you can't make proteins. So when you consider how important proteins are generally, not being able to make them would be pretty disastrous. Now, one last thing to look at, in terms of the animal cell anyway, the centriole. Now, these things are tubular structures arranged sort of, well, in a cylinder, packed in units of three. In fact, if you like, have a close look at this electron micrograph, and you can probably just about see the sort of the threesome structure. Now, the protein that makes those up is, very imaginatively, called tubulin. Now, without these things, you wouldn't be able to get any cell division going on, because during mitosis, these things pair up to create centromeres, which then produce the aster and the spindle that move the chromosomes around. Chloroplasts, we'll start with those, shall we? As we already know, chloroplasts are where photosynthesis happens inside a plant cell. Something you also already know is that they contain a green pigment called chlorophyll, and that kind of makes photosynthesis able to happen. Now, what you don't know yet is the structure of the organelle itself. Here we go then. Right, your chloroplast is made of three membranes. You've got an outer one, you've got an inner one, and you've got the business end, the thylakoid membrane. Now, the thylakoid contains two types of chlorophyll, and biochemists, being so imaginative, have called them chlorophyll A and chlorophyll B. Now, it's those molecules getting excited and releasing electrons that gets the whole photosynthesis process going, and we'll talk more about that. Guess when? Yep, in a later video. Now, in order to expose the maximum amount of chlorophyll to sunlight, your thylakoid is packed really tightly into stacks that are called grana. And each of these grana are linked to each other by stromal lamellae. So, like, really, kind of like passages between one and the other. What you've got is one big thylakoid membrane that is folded and folded and folded and folded into these grana and the lamellae. And to finish off, the vacuole. Now, in plants, it's big area, pretty much in the middle of the cell, surrounded by a membrane, lipid bilayer, and it's full of cell sap. So, basically, glucose and water. Feeds the cell and provides a bit of strength. In an animal cell, you do get vacuoles, but they tend to be a lot smaller and temporary. And it's short-term storage, so starch, things like that. Okay, we'll bring this one to a halt. What have we learned? We've learned two things, really. There are two types of cell, prokaryote and eukaryote. We've also learned that eukaryotes have a number of organelles, each bound by a membrane and with a specific job. Prokaryotes have no internal membranes, have an area with nuclear material in them, but other than that, everything is handled by the cytoplasm. And that, my friends, is a high-speed tour through cellular biology. And we've only just got started.